Hello everyone. So in the last session we are discussing about the 5 kingdom system proposed by R.H. Whittaker in 1969, and he gave a better combination, a better characters based on those he identified, or he proposed 5 kingdom classification. So the 5 kingdom classifications are: first one is kingdom Monera, next is kingdom Protista, the next one is kingdom Fungi, fourth one is kingdom Plantae, and fifth one is kingdom Animalia. Like this he proposed 5 kingdom system. Nowadays we are following the same, and so I put one table to make you understand how the characters, how based on those characters how he separated the organisms. And the next thing is, and today, it means today in this session we are going to start with the first kingdom, that is kingdom Monera. Okay. Kingdom Monera. The kingdom Monera, it consists of, as I told you in the last class itself, it includes the bacteria, number of abundant bacteria found on the earth. Some of them are useful and some of them are pathogenic. So some of them not only harmful or not only pathogenic, useful or harmful, except these character, some bacteria are there known as Archaeobacteria. They live in harsh or extreme environment or in extreme habitats like hot springs and in the gut of the cattle, cow and buffaloes guts. The bacteria are there. Those bacteria live in the gut of the cattle, and some of them are highly acidic. Highly acidic. Some of the bacteria lives in a high acidic environment, and some of them are highly thermal, resistant to high temperature. So these are also present, which comes, and this type of bacteria are there which comes under kingdom Monera. Now my dear students, first we'll look at the different types of bacteria based on the shapes. So one by one I'll tell you how the bacteria are there, how they differ from one another. Number of bacteria are there, number of thousand millions of bacteria are there. Some of them are useful to human beings, some of them are harmful to not only human being but also to plants. So they causes diseases to plants, animals, humans, and also some of the bacteria infect the bacteria itself. So we'll list out one by one, and with that we also study, we will study about the Archaeobacteria. What about the Archaeobacteria? How we can define or we can describe the Archaeobacteria. So first of all, I'd like to mention here the different types of bacteria based on the shapes. Based on, so whenever they'll ask you to write the different type of bacteria based on the shape, you have to write this answer. So the question will be for 2 marks. If I conduct that test for the unit test, I'll ask to write with diagram. I'll ask you to write the question. I'll ask you to write the answer for 3 or 5 marks. Okay. So if the question arises in unit test, it will be for 5 marks. Okay, but in the board examination, final examination, the weightage of the chapter is 2 marks. Based on the weightage, they'll ask the question. So different type of bacteria based on the shapes. They have given based on the shapes. Like different shapes of bacteria are there. The shapes are, one is spherical, So this is one spherical bacteria, and one more. Like that, different number of bacteria are there and their shape is spherical in nature, and that type of bacteria is known as cocci. Okay, so this is the first type of bacteria, and the second type is rod shape. So these are spherical bacteria, spherical shaped bacteria known as cocci. This is the shape name and this is the bacterial name. And here one more they have given in your NCERT book. The name of this bacteria is Bacillus, and the shape of the bacteria is rod shape. Example, Bacillus, rod shape bacteria. And the third type of bacteria is spiral shape, so this is one bacteria, a spiral shaped bacteria. So these are the flagellas. Okay, and the name of the bacteria is spirillum. So the bacterial name is spirillum, the shape is spiral. And here I will write the fourth one. The fourth type of bacteria is comma shape. Comma shape. Okay, so these are also type of bacteria which look like a comma shape, and the example it means the name of this type of bacteria is Vibrio and the shape is comma shape. Okay, so you have to remember all the 4 types of bacteria divided based on the shapes of shape. Okay, so next is, after this concept, first what we studied, so we are in the kingdom Monera. The Monerans, it means the Monera organisms. Moneran species include maximum bacteria. The bacteria are unicellular and prokaryotic. Don't forget this point. Unicellular and prokaryotic. Some of the bacteria are useful and some of the bacteria are pathogenic, and some are live in extreme habitats. And after that we are going to start the next concept. It means the different type of bacteria based on the shapes. So first type of bacteria, so the shape is spherical. The name of the bacteria is cocci. The shape is rod shape and the name of the bacteria is Bacillus, and the shape is spiral. The name of the bacteria is spirillum and the shape is comma shape and the name of the bacteria is Vibrio or Vibrio. Okay, so we'll move on to the next concept. The next is, here I just wanted to give a brief description about the Archaeobacteria. So here concentrate on this. Don't forget we are in the kingdom Monera. Alright. So we have completed studying the types of bacteria based on the shapes. And the next thing is we are studying about the Archaeobacteria or ancient bacteria. So the Archaeobacteria, what is much important, what is the special feature present in the Archaeobacteria? So the Archaeobacteria lacks peptidoglycan. In the cell wall, it lacks the peptidoglycan. Pseudopeptidoglycan type of chemical component is present in the cell wall of Archaeobacteria, so that they can resist to high temperature or highly acidic content, or in the highly acidic habitat they can live. So why they are more resistant? Why it is known as ancient bacteria? Because they have the resistance capacity to survive in extreme habitats. Okay, so I'll list out some of the Archaeobacteria found in different habitats. Alright, one is Halophiles. Why we are going to name the bacteria Archaeobacteria like Halophiles? Because these are the Halophiles, or these are the type of Archaeobacteria found in extreme salt region, extreme salt areas. Extreme salt areas. Okay, one is Halophiles. So they used to ask in the examination, what is Halophiles? Halophiles are the type of Archaeobacteria found in extreme salty areas. So that is the answer. Extreme salty areas. The salty areas in sense this is the water media. In the water media, where do you find the extreme salt areas? It is nothing but the oceans. In the oceans or deep seas or deep oceans where the salt content is more, there the Halophiles are appear. The Halophiles live. Okay. So the next is Thermoacidophiles. Write down Thermoacidophiles. Lives in hot springs. Where they find? So the Thermoacidophiles, if they ask you to write about the Thermoacidophiles, what is Thermoacidophiles? Thermoacidophiles are the type of Archaeobacteria found in hot springs. Hot springs means in high temperature they have the resistance capacity. They have the resistance power or resistance capacity, so that they can survive in high temperature or hot springs. And third one is Methanogens. Methanogens, yes, these are the number of times they asked about the Methanogens. You have to observe this Methanogens now. The Methanogens are the type of Archaeobacteria found in the gut of the cattle. Very simple how to understand the Methanogens. You might have heard about the methane gas or biogas. How they prepare, how they produce the biogas. Some of them it has it will be having a tank, underground tank, and to that tank cow dung slurry will be passed there. After the fermentation of the cow dung, one gas releases. That gas collected through the pipes. It will be supplied to the nearby houses, and by using those biogas, automobiles and it is used for the cooking purpose, and it is used for the automobiles. So biogas has multi-purpose. It is used for the multi-purpose. Okay, so how they utilize? So what is the primary raw material for the production of biogas? What is the reaction takes place there in the tank? How the biogas releases? There the question arises. So whatever we know to our knowledge, just we know the cow dung is collected and passed into the tank. The cow dung, what I mentioned here, the cow dung, cows or buffaloes dung should be collected. Okay, so concentrate on my words. In the cattle, the gut of the cattle, cows or buffaloes, methanogens are present. Methanogens are the type of Archaeobacteria where it lives in the, you can understand in the gut like a digestive tract, there number of enzymes will be present, number of acidic content will be present. In that acidic content, the methanogens are present. When it passes through the dung, where it comes out through the dung, if the dung is utilized for the production of biogas, there in the dung, in the fermentation process, methanogen plays a very important role. It oxidizes or it converts the number of chemicals into methane. That methane used for the multi-purpose. It has been used as the next step. Okay, so here they'll ask you about the methanogen, how it is utilized or where it is present. What is methanogens? Methanogens are nothing but these are the type of Archaeobacteria where it is used for the production of, or it is responsible for the production of methane gas or biogas. Just write down as biogas. Okay, so these are the three different types of Archaeobacteria where based on their habitat they have given the three different bacteria. So that's all about the Archaeobacteria. Now we'll move on to the Eubacteria. The Eubacteria are true bacteria. It has a rigid cell wall. It's not like Archaeobacteria doesn't have a rigid cell wall, that also have a rigid cell wall, and the Eubacteria is known as true bacteria. Eubacteria are true bacteria. And second one is it have a rigid cell wall. If it is motile, it consists of flagellum. Flagellum means just now I wrote one type of bacteria like spirillum. I wrote here, no, it consists of flagella, number of flagella over here and there. What is the use of flagella? It helps in the locomotion. If it is a if the Eubacteria locomoting from one place to other place, it will be with the help of a flagella or flagellum present either at both the ends or towards one end. Okay, So the next is true bacteria. These are consists of a rigid cell wall. If it is a motile, it will be having the flagellum. Okay, so these are the very basic characteristic features of bacteria. So more than this, the very important thing what I am going to write now. So this is important. Photosynthetic autotroph, chemosynthetic autotroph, and the one more is heterotrophs. Chemosynthetic autotrophs, photosynthetic autotrophs, heterotrophs. One more is there, that I'll mention you a little bit later. So I have divided the eubacteria into 3. On what basis I have divided into these 3? Based on the mode of nutrition, whether they are producing their own food or not. Based on that, I have divided into 3. It is not I divided these according to your NCERT syllabus. So first one what I am written here is photosynthetic autotrophs. Let me tell you the many meaning of autotrophs. Autotrophs means self preparing the food. So I used to prepare my food. Do you call me as an autotroph? No, we are dependent on other organisms for the food and nutrition. Some of the bacteria are there, some of the algae are there, some plants are there. They prepare their own food with the help of by using sunlight or radiation. So those are known as autotroph. Here bacteria are also known as, some of the bacteria are also known as photosynthetic autotroph. Why so? Those, let me tell you one example here. Let me write one example first. Cyanobacteria. Again it consists of number of organisms like cyanobacterians. Example, Nostroke. It's not Nashta, it's Nostroke. One more is B-9. So these two examples, these two bacteria comes under photosynthetic bacteria, photosynthetic autotrophs comes under the true bacteria or eubacteria. Don't forget, we are in the kingdom Monera. Monerans, we studied about only few features we studied about the bacteria or kingdom Monera. In kingdom Monera, maximum of the bacteria, maximum of the organisms are the bacteria. The bacteria can be differentiated into 4 types based on the shapes. Okay, so one is Pocci, which is spherical in shape. Second one is rod shape, bacillus. Third one is spiral shape, spirillum, and one next is comma shape, vibrio. So 4 different shapes of bacteria we studied. After that, we had a brief account on the Archaeobacteria. Based on the habitat, Archaeobacteria has been classified into 3. One is Halophiles, Thermocytophiles, and Methanogens. After that, we are in the eubacteria. Eubacteria are also known as true bacteria. They have a rigid cell wall, and if it is motile, it consists of a flagellum. Based on the mode of nutrition, again the eubacteria can be classified into 3 types. One is photosynthetic autotrophs, chemosynthetic autotrophs, and heterotrophs. In the photosynthetic autotrophs, the very first, the common example is Nostroke and Anabina. Okay, so how they prepare their own food? Unlike photosynthetic autotrophs like blue-green algae, how the algae produces their food, unlike that, similar to that, photosynthetic autotrophs also prepares their own food. Okay. Now, the next is, let me introduce you, tell the characteristic features of Nostroke here. The Cyanobacteria are, or the Nostroke or Anabina are unicellular. Let me was remembering this. Okay. Important. They consist of chlorophyll A pigment. They are inside the cell. Why they are preparing the food? Because they are not plant. Only plant can synthesize their food by using sunlight, right? So that we know. But in the kingdom Monera, bacteria, some of the bacteria are also preparing their food with the help of sunlight. How? Because of the presence of this pigment, chlorophyll A. Because of the presence of chlorophyll A, they prepare their food. Okay. So now I am going to describe about the Nostroke. Now Nostroke is unicellular, filamentous, colonial organism, colonial bacteria, and the cells are arranged one above the other. The cell wall is, or this Nostroke filament is covered by a gelatinous sheath, and it has a special cell known as heterocyst. With the help of the heterocyst, they synthesizes their food, heterocyst. Okay, so special cell known as heterocyst. Alright, and they often form blooms on the polluted water bodies. They forms the blooms like if it is a water body, polluted water body, they forms this kind of blooms, and this Nostroke help in the fixing of atmosphere nitrogen. Don't worry if you are not getting about this. I'll make you understand again. It will come in the next upcoming preceding chapters. There I'll make you understand. Just concentrate on these features. Let me draw the diagram of Nostroke so that you'll come to know how the Nostroke looks. Here I'm going to draw the structure of Nostroke. Don't consider this is the chemosynthetic bacteria, this is the photosynthetic bacteria. Okay. Now I am going to label the diagram. Just complete this diagram. Now, let me tell you the Nostroke, about the Nostroke. The Nostroke, as I said, unicellular, filamentous, filaments, like here one cell, one more cell, one more cell, one above the other, one above the other, they form into filament like this. Number of Nostroke filaments grouped in one place. So that group of filaments of Nostroke can be known as colony. They'll present in the colonial form. By forming a colony, number of colonies, they will be present in the substances or on the water body or in the polluted water bodies. So that is the first case feature definition or the description. They are covered by this filament of the Nostroke covered by gelatinous sheath. So here the gelatinous sheath will be covered throughout the filament. One filament is covered by gelatinous sheath, one more is covered by gelatinous sheath. Number of like this, all the filaments of Nostroke covered by the gelatinous sheath. So these are the cell, and the cell will be having the cytoplasm and nucleoid. So like this, the cell organelles will be present. Okay, so here a horseshoe shape one cell is present like this. Let me draw here. So this is one cell. Inside the cell, it consists of a special feature, a unique feature, a horseshoe shape cytoplasm is present. So just while describing the heterocyst, while writing this feature, I told they synthesizes the food, but make it correct. The heterocyst fix the atmospheric nitrogen. Some of the cells, they will be having a horseshoe shaped structure. Those special cells are known as heterocysts. Those heterocysts fix the atmospheric nitrogen, and they form, like if they arranged on the water body, polluted water body, if the number of filamentous Nostroke form on the water bodies, they look like a bloom. Okay, so it's all about the Nostroke or Nostroke. These are the features of Nostroke. It's not about the Anabina, and they comes under the photosynthetic autotrophs. Now let me tell you about the chemosynthetic autotrophs. Chemosynthetic autotrophs they oxidizes various organic or inorganic substances like nitrates, nitrites and ammonia. It oxidizes various inorganic substances like nitrites, nitrates and ammonia. Okay, so this is the first feature of chemosynthetic bacteria. What is the chemosynthetic bacteria? I'll tell you in short. I'll tell you, because what is the purpose of writing the feature of chemosynthetic bacteria? My concept is the purpose, like the function of the chemosynthetic bacteria. What is the first function? They are going to oxidize the inorganic elements known as nitrite, nitrate and ammonia from the from by utilizing the energy. When they oxidizes these kind of organic substances, they will get some energy. By utilizing that energy, they produce adenosine triphosphate. So when they oxidizes these kind of inorganic substances or this kind of substances, it will be useful to prepare the ATP energy, which will be utilized for the production of energy, for the production of proteins and all inside the body. So this is how they are not dependent on other organism. By oxidizing various substances, they are preparing their own food, not utilizing the light energy but utilizing some chemical substances. So this is how chemosynthetic autotrophs work, and they play a very important role in recycling the nitrates and nitrites and nitrogen, phosphorus, iron, sulfur, nitrogen, phosphorus, iron, sulfur. So these are the substances. If it is present in the soil, it increases the fertility of soil. The thing is, how the soil fertility increases, if it has these kind of substances in the soil. How these kind of substances enter inside the soil? By the oxidation of these type of bacteria, directly or indirectly, these useful substances enter inside the soil. When it enter inside the soil, it increases the fertility of soil. And if the soil fertility increases, what is the benefits of it? No doubt, the crop of the plant, the yield will increase. If the soil fertility increases, the plant grow faster and it yields more. So this is how these type of bacteria are very useful. They are going to recycle the nitrogen, phosphorus, iron, sulfur type of substances. So this is also you have to remember important one. Okay, now coming to the heterotrophs. Heterotrophs are dependent on other organism for their nutrition. So next is how they are important, how they are significant, in what field they are significant, any useful bacteria are there in heterotrophs. Yes, exactly. In our daily life, we used to consume curd, or we used to have the antibiotics if we fell ill. What the doctors give us? The doctor give us some antibiotics. Not all antibiotics prepared by the bacteria. Some of the antibiotics prepared by the bacteria. Okay, so the preparation of antibiotics, conversion of curd from milk, and in the preparation of alcohols, or in the preparation of fermentation, in number of fields, the heterotrophs bacteria are utilized. So this is the useful bacteria, and this type of bacteria are abundant in nature. Number of bacteria are available in nature, and some harmful bacteria are also there. Harmful bacteria in sense they are going to harm, they are causing diseases not only to human beings but also to plant crops and to animals and to the humans and so on. So, useful bacteria, they can be utilized for multi-purpose. Multi-purpose means in kitchen also we used to use the bacteria, useful bacteria, helpful bacteria, and some of the bacteria are causing diseases. Some of the bacteria are pathogens, not only to human beings but also to plant crops, animals, humans, dogs, cats, like that. Okay, so how they reproduce? So if we come to the discussion of reproduction, how the bacteria reproduce? The bacteria reproduce by means of asexual and by means of formation of spores and also by means of sexual, asexual, sexual and by means of spore production. Let me tell you in detail about the asexual mode of reproduction and the sexual mode of reproduction and how it forms a spore. Asexual mode of reproduction forms by means of, it occurs by means of binary fission. Okay, so let me write here binary fission. The binary fission, so this is the type of cell division where the cell, consider this is the rod shaped bacteria. Okay, so when it has to divide itself, when it has to reproduce, when it has to multiply its number, what it will do? Simple it is. It forms a cleavage at the middle of the bacteria. Before the formation of cleavage, what it will do? It will divide. The DNA will replicate. Only one DNA will be there in the bacteria. Okay, when it comes to the division, if it is more enough, if it is mature enough to divide, at first primarily what it will do, it will divide its DNA first and all the organelles will divide equally. So if the division takes place in the DNA, one DNA will be here and one more DNA will go to the extreme end and all the organelles will be present this side. And after the division of DNA, the cleavage occurs. That cleavage separates the DNA into two equal halves. So that is known as binary fission. Without the involvement of two parents or without the involvement of gametes, thus the reproduction took place in the bacteria. This type of bacteria is known as asexual. This type of reproduction is known as asexual reproduction, where the one cell give rise to two equal halves. So this is the small DNA now, so small bacteria now. Now this bacteria will grow and mature, develop. The type of asexual reproduction is binary fission. What is binary fission? So binary fission, this is the type of asexual reproduction where the cell reproduces and it gives rise to two equal daughter cells. So these are two daughter cells now. Okay, now sexual reproduction. Sometimes the bacteria, consider this is one bacteria and one more bacteria will be there. So this bacteria, sometimes this bacteria, so this is the type of sexual reproduction I am telling you. This bacteria, it will transform the DNA, its DNA to the surrounding DNA. One bacteria, it will first it will forms a bridge in between these two bacteria, if they want to transform, if they want to reproduce, what the bacterias, what I am telling you about the sexual reproduction takes place in the bacteria. So the bacteria also reproduce by means of sexual. After transforming the DNAs from one bacteria to other bacteria, one bacteria to other, after transformation, consider here one DNA will be present, here one DNA will be present. Okay, at first they form a bridge, and then this bacteria transfer into one bacteria, and here it will packed with this DNA. Again the packed DNA will transform into one more bacteria, and the bridge dissolve, and then they divide. So this is how they form, they transfers the DNA from the subsequent from the one more bacterium. Only here the DNA transforms, and then the bacteria divide. So we can call this type of reproduction as sexual reproduction, because they are crossing the one more some other bacteria's DNA. That's why we cannot call this as asexual, we can call this as sexual mode of reproduction. Okay, so consider one more bacteria. What I am telling you here I am telling you about the spore production. So if it is in an unfavorable condition, the condition is the environment is the ambience is not suitable for it. What it will do, it doesn't reproduce, it will not the metabolical changes will not take place inside the cell. What it will do, it will divide its cell, not completely, it forms a cell, there it forms a spore, okay, and then this is the mother cell, and this is the spore. All the useful materials engulfed by the spore, and then this spore will be remain at the central position, and it will be like that for years ago, till it reaches to the favorable condition. All the metabolic condition, all the metabolic activity or the reproduction will be ceased, and then when it reaches to the favorable condition, when it has a favorable suitable media, then the spore releases out. The spore comes out from this cell wall, and then they reproduce, they grow, develop, reproduce, and the normal activity will take place. So this is how the spore production takes place. Only one example I explained here. Number of different spore production examples are there. Mode of the methods are different. Only simple method I have taken here. Spore production, the description is during unfavorable condition, the bacteria forms the spores, and when it reaches the favorable condition, they releases their spores, and the normal activity takes place. And the last one is Mycoplasma. Mycoplasma, these are the bacteria, they lack cell wall, and they are living and smallest bacteria. So many times they asked you in the examination, what is mycoplasma? Give a brief account on mycoplasma. What you will write? Mycoplasmas are the type of bacteria, or the bacteria where in this bacteria the cell wall is absent, and then they are living and smallest bacteria. Compared to other bacteria, the mycoplasmas are very smallest bacteria, and the mycoplasma are pathogenic, highly pathogenic. They are disease causing bacteria. They causes diseases in plants and in animals. Okay, so we have completed the kingdom Monera. Now, let me tell you one more thing about this Mycoplasma. The Mycoplasma is the bacteria where it lacks the cell wall. It is a living bacteria and smallest bacteria, and it can survive without oxygen. One more feature is the Mycoplasma can survive without oxygen. Okay, so let me tell you one more thing, make it correct. The Nostroke, I told you, no, Nostroke, I wrote gelatinous sheath. It can also known as mucilaginous sheath. Mucilaginous sheath. Only that much you have to. One more name of the gelatinous is mucilaginous. Okay. So that's all about the kingdom Monera. If you have any doubts, please ask me. Thank you.